Celiac disease is an immune-mediated small intestinal disorder triggered by the consumption of gluten in susceptible individuals. The characteristic immune response seen in celiac disease can precipitate a host of debilitating symptoms. Currently, the only method to prevent celiac disease is strict adherence to a lifelong gluten-free diet. However, this proves challenging for many due to the inaccessible nature of gluten-free products. Additionally, an increase in demand for Coeliac-Safe wheat-based products has led scientists to research whether the gluten content of wheat can be modified via genetic engineering. Genetic technologies of interest in this process have included RNA interference, or RNAi, and the CRISPR-Cas9 system. Although both technologies have their drawbacks, they have successfully attenuated the immunogenic nature of wheat for patients with celiac disease, providing promising results for the future.
In order to understand the physiology of celiac disease, we first need to understand the process of digestion and the form and function of the small intestine, the biochemical properties of gluten, and some of the key features of the human immune response. Digestion begins in the mouth, where enzymes such as amylase catabolize the starch content of food, and our teeth manually break down the food to increase its surface area. After swallowing our food, it passes through the esophagus and lands in the stomach. Food is further broken down into smaller particles in the stomach's acidic environment. The process is aided by enzymes such as pepsin, which catabolizes proteins. Once the food is sufficiently broken down into smaller particles, it passes through the pyloric sphincter into the small intestine, a 22-meter-long organ where nutrients are absorbed. The upper portions of the small intestine further process food into its final form, so that it is ready for absorption. The small intestinal mucosa, or lining, contains millions of villi, made up of epithelial cells. Villi are finger-like projections responsible for the absorption of food from the small intestine into the bloodstream. The curved projection-like form villi take on allows for them to efficiently take up nutrients via both passive and active transport.
What is it about gluten that upsets the small intestine so much, to the point an immune response is elicited? To answer this, we need to uncover some of the biochemical properties gluten possesses. Gluten is a highly complex compound, predominantly composed of gliadin and glutenin proteins. Gliadin and glutenin proteins both contain high amounts of glutamine and proline, which are amino acids that are particularly resistant to proteolytic digestion by proteases in the human digestive tract. That is, these amino acids prevent gliadin and glutenin proteins from being catabolically digested by the enzymes that break down proteins. Gliadin is known to be particularly toxic to epithelial cells of the small intestine, and it is thought that the incomplete digestion of this protein contributes to the immunoreactivity of gluten in coeliac disease. The final piece of knowledge we need to understand coeliac disease is how the human immune system works. Little white blood cells called macrophages float around the body looking for foreign materials. When the macrophage encounters something foreign, it engulfs it and digests it via phagocytosis. Once this foreign material has been digested, the macrophage displays the leftover fragments on its surface on an MHC2 marker. The macrophage shows this fragment to a T helper cell, and the T helper cell triggers the release of chemicals called cytokines. Some cytokines directly destroy the foreign material, other cytokines trigger other cells to destroy the foreign material. For example, B cells are responsible for secreting antibodies to inactivate the invading substance.
So what is it that makes someone susceptible to celiac disease? From a genetic perspective, it's thought that the human leukocyte antigen genes, HLA-DQ2 and HLA-DQ8, contribute to the development of celiac. From an environmental perspective, a range of factors have been posited to contribute to the disorder, such as intestinal dysbiosis, which is an imbalance of good bacteria and bad bacteria in the gut, leading to abnormal immune system activity. However, it unfortunately still remains somewhat ambiguous as to what makes someone susceptible to the disorder. When we consume gluten-containing products such as bread, the food is digested normally, up until it reaches the small intestine. The incompletely digested gliadin and glutenin peptides found in gluten become elongated and are able to make their way into the intraintestinal space. The presence of the gliadin and glutenin peptides in this intraintestinal space leads the body to mount an immune response in patients susceptible to celiac disease. In this immune response, epithelial cells send out distress signals, leading to the release of cytokines. Macrophages are also instructed to assemble in the small intestine to degrade the seemingly foreign gluten compound. Gliadin and glutenin fragments attach to the HLA-DQ2 and HLA-DQ8 MHC markers on the macrophage. The macrophage presents these fragments to T helper cells, which instruct B cells to proliferate. B cells produce antibodies which target gliadin peptides specifically. However, these antibodies also target tissues of the small intestine. The immune response can precipitate harmful structural changes in the small intestine, such as villus atrophy. Villus atrophy results in the villi becoming flattened, hence they cannot effectively absorb nutrients. Consequently, celiac disease can result in a range of other symptoms, such as nutritional deficiencies, osteoporosis, infertility, abdominal pain, diarrhea, and vomiting. The debilitating nature of celiac disease, a lack of accessible gluten-free products, and growing demand for celiac-safe wheat-based products has led researchers to investigate the use of genetic technologies to modify the gliadin and glutenin contents of wheat. Unification refers to the process of altering the genome at specific target locations. RNAi is one technology that's been used to modify the gluten content of wheat. RNAi indirectly targets genes by interfering with their mRNA transcripts. RNAi works by first synthesizing an RNA molecule designed to bind to the mRNA transcript of the target gene. The synthetic RNA molecule is integrated into the cell of the to-be modified organism, and it latches onto the mRNA transcript of the target gene. In binding to the mRNA transcript, mRNA translation is inhibited, thereby silencing the expression of the target gene. A series of studies have used RNAi technology to downregulate the expression of several gliadin proteins in wheat by up to 85%. Although this technology has been successful in reducing the immunogenic nature of wheat for patients with celiac disease, it often produces significant off-target effects.
The CRISPR-Cas9 system was first characterized as an immune system for bacteria and archaea.
The observation that the CRISPR-Cas9 system can remove segments of genetic material in prokaryotes led scientists to engineer the system to serve as an RNA-guided nuclease for genomic modification. For in vivo genetic modification using CRISPR-Cas9, synthesis of a single guide RNA or sgRNA molecule with complementary bases to the target DNA sequence is required. A Cas9 nuclease is bound to the sgRNA and the complex is integrated into the nucleus of the to-be modified cell. The synthetic sgRNA molecule guides Cas9 towards the target gene locus, where the nuclease creates a double-strand break. Disruption of the double strand triggers the activation of non-homologous end joining, or NHEJ for short, which is an error-prone innate DNA repair mechanism. NHEJ results in nucleotide insertions and deletions, indels, at the target sequence, rendering the target gene dysfunctional. Sanchez-Leon and colleagues used CRISPR-Cas9 to modify the gliadin content of wheat in 2018. They synthesized an sgRNA molecule with complementary bases to the genes encoding for gliadin proteins in wheat. The RNA Cas9 complex successfully mutated the target gliadin genes, producing a line of wheat with 85% less immunoreactivity than traditional wheat. Additionally, no off-target effects were found in the modified wheat, demonstrating the high precision of CRISPR-Cas9 for genetic engineering.